Alright, look at the big screen, everyone. As Dr. Strawberry, someone so legendary that even caterpillars stand at attention when they see me. Today, I wanna share you a top secret. We, strawberries, are not true fruit. Shocked? Amazed?
What? What?
That juicy red part you devour is actually an elite accessory fruit. It is a botanical masterpiece created by the magnificent expansion of my receptacle. And those tiny black dots on my skin? Don't you dare look down on them. Those are my actual fruits, known by the elegant name achenes. Each little achene cradles a microscopic seed of destiny inside. As masters of reproduction, we are incredibly versatile. We don't just rely on seeds for sexual reproduction, but we can also stretch out our proud stolon for asexual cloning, multiplying my flawless excellence everywhere. Now, look at this radiant crimson complexion that makes everyone fall in love at first sight. This isn't cheap blush, darling. It is premium and anthocyanin. These molecules aren't just my gorgeous royal wardrobe. They are high-powered antioxidant shields protecting us from UV radiation. Of course, if you want to pamper a VIP like me, your environmental standards must meet royal specification or we will straight up go on a strike. We thrive best in temperate, subtropical, or marine west coast climates. Want us to germinate? Keep the temperature strictly between 20 to 25 Celsius. During our growth spurts, we demand 18 to 25 Celsius by day to shine and 10 to 15 Celsius at night for our beauty sleep. When it's time for the grand final fruiting, the sweet spot is a perfect 15 to 22. For sunlight, you must serve us 6 to 10 hours of moderate intensity light daily. Nonetheless, water management is where you really need skills. An annual rainfall of 600 to 800 millimeters is ideal, with ambient humidity perfectly maintained at 60 to 80%. Finally, the mattress I sleep on must be top tier. We only accept premium loam or sandy loam with a soil moisture level of 60 to 80% and a meticulously balanced soil pH level between 5.5 and 6.5. Get all that? That is the ultimate guide to raise a genius like me, Dr. Strawberry. Since I'm already here, let me tell you another ultimate biochemical secret behind our growth spurt. Listen up. The true mastermind boosting this growth spurt is called indole-3-acetic acid, IAA. Don't let the name scare you. It is the most core, premium natural oxime inside the plant. However, as powerful as IAA is, it doesn't just appear out of thin air. Just like how my dashing PhD look require an exquisite touch of makeup. Supreme intellect. It requires an indispensable soulmate of an biochemical precursor, L-tryptophan, which we call LTRP. Inside our meristems, L-tryptophan walks down a red carpet called the tryptophan dependence pathway. Along the way, it passes through strict review by enzymes catalytic officials like IPA and TAM, before finally making a gorgeous transformation into physiologically active IAA. Want our root systems to explode with growth, flower buds to pop like crazy, and fruits to be sweet enough to rot your teeth? Exactly. That is the true essence of my experiments this time. Remember to use the perfect LTRP concentration dosage effects to completely master our destiny. Thank you everyone. You may stop applauding now.
Thank you, Dr. Strawberry, for that memorable entrance. You certainly have a flair for the dramatic. But now, let's take a look of what happens behind the scenes of our project. As Sophia just revealed, IAA, this famous auxin, is the ultimate enzyme driving the plant. In plant science, we know that auxin is essentially a superpower. It stimulates robust root development to maximize water absorption. It optimizes internal vascular bundles, the xylem and phloem, to transport nutrients. It also signals the fruit to grow larger after pollination and prevents premature flower and fruit drop. In theory, auxin is magic, but our project takes a step further. We want to test the exact limits of Dr. Strawberry by introducing its precursor, L-tryptophan, at different concentration.
Oh my God! Wow!
Our hypothesis is that the plant's response to this auxin boost won't be a straight line, but will follow a dose-dependent curve. Think of it as biological optimum. There is a dose that is too low, a dose that is just right, and an absolute overdose. Here is what we expect to see. At 1 micromole, it's the gentle boost. At this low dose, auxin will mostly work backstage. We expect it to enhance root development and vascular efficiency, giving the plant a subtle upgrade. Visually, this should translate into longer stolons and larger leaf surface areas compared to our control group. At 3 micromole, it's the sweet spot. This is where we expect the real fireworks. This optimal auxin level will maximize cell division and strengthen the flower stalk. The predict result? Reduced flower drop, significantly larger and redder fruits, and a major boost in commercial value and sweetness. At 5 micromole, it's the overdose. In biology, too much of good thing is a bad thing. Sating the plant with this high concentration will trigger the production of ethylene, a major stress hormone. Instead of helping, it will stunt root growth, cause leaf epinesty, and accelerate plant aging. This experiment is fascinating because it connects pure biochemistry with real-world agriculture. If we can prove that a specific dose of L-tryptophan can maximize fruit size and stability without triggering an hormonal strike, we could provide farmers a game-changing, eco-friendly method to boost crop yields.
I love it! Come on, let's go!
So, will Dr. Strawberry achieve ultimate perfection, or will she end up completely stressed out? Place your bets.
Hello everyone, I'm Robert. My parts are conducting the experiment and recording the results. Let's get started. Due to persistent rainfall starting in mid-April, our experiment was temporarily stopped as environmental variables became uncontrollable. Sadly, 4 of our plant samples progressively wilted and perished due to waterlogging caused by the continuous rain. It was not until early May when the weather cleared up with abundant sunlight that we concentrated our experimental trials over a critical 3-week period. Our experimental design consisted of 3 groups: a distilled water control group, a 1 micromolarity tryptophan treatment group, and a 3 micromolarity tryptophan treatment group. To prepare the solutions, we utilized an electronic analytical balance to measure the powder weight, tiered the device, dissolved the chemical in distilled water, and stir the mixture until fully homogeneous. This routine, consisting of controlled watering and solar illumination, was carried out daily from May 11 to May 29. Concurrently, we monitored and logged daily environmental metrics, including temperature, relative humidity, and ultraviolet intensity. As demonstrated in this chart, the ambient temperature throughout May was exceptionally high. Temperatures exceeding 30 Celsius became the norm, with the peak temperature reaching as high as 34.7 Celsius. Furthermore, the ultraviolet index exceeded a critical threshold of 10 on 5 separate days. Both extreme heat and high ultraviolet radiation represent severely adverse environmental stressors for strawberry growth. Regarding relative humidity, apart from a few days where it surged to 99%, the levels remained relatively stable and within a normal range. Here are our data logs and photographic evidence, all for the ambient temperature. During these 3 weeks, we observed that the substrate in most parts remained severely water-saturated. The soil surface was colonized by algae, bryophytes, and opportunistic weeds. Furthermore, the leaves and stems showed clear signs of severe wilting and necrosis, while the root systems exhibited apoptosis and decay. Only parts number 3 and 4 managed to retain green leaves. However, upon examining the root architecture, we noticed that the roots of part 3 had already undergone significant browning and necrosis. Sample 4 was the soil sample whose roots retained a substantial amount of viable, healthy, white tissue, the only one.
Next, we're proceeding to the discussion section. I am Vivian. And hereupon is the outline for this section. I will walk through experimental data, key findings, advice mechanisms, and hypotheses with literature support. So here's the weather context from our experimental duration from May 12 to May 29, which we observed that continuous rain has saturated all 6 plots. Moreover, critically, rain has been falling for 2 weeks before the experiment began, which is the arriving season of plant rain. Lander weather shifted. May 24 to 28 brought a heat wave that rose to 37 to 38 degrees Celsius with extreme UV level. The rain then heat sequence is critical to the plant's survival story that we are going to tell later. Then we're gonna go through our endpoint measurements. So this is our actual data on the final day of experiment duration, which is May 29. The key pattern we see is that 4 of 6 plants died. All 4 were inside. Both outside plants survived. We could also observe how these dead plants share similar features, with blackened roots and withered leaves. As for stem lengths, across 7 days, we noticed that only 1 millimolar plant P3 and P4 grew by 3 to 4 centimeters, and both control and 3 millimolar groups stayed nearly flat. So here's the plot. This pattern we could see that peak effect at moderate dose but cancel at high dose. This is the signature behavior of the IAA pathway. Then is the soil moisture and moss. Moss grows when the soil never dries, so it's actually a key indicator of soil moisture. And 3 centimeters of moss in dead plots mean weeks of saturation, only 1 centimeter in the healthy P5 group. Next slide, we go through leaf state. We can see the gradual leaf yellowing in this plot except for P5. And stem rigidity plot. The condition deteriorated just like how leaf declined. However, P5 appeared again to be more rigid this time, as well as P4. So here's the key findings from the direct observation of our data. Following the data observed, our discussion revolved around 3 questions. The first is root cause of plant death, and the second is the effect of L-tryptophan on plants, and the third is other observed mechanisms. So we can actually conclude in 3 directions. First, survival track position, not treatment. Second, soil moisture is the strongest correlate of root death. Third, stem elongation show clear tryptophan dose response. So here's the first hypothesis that we propose: IAA hormesis curve on stem elongation. Here's some background. IAA is the main natural auxin in plants, the hormone that drives cell growth. Tryptophan is the precursor of IAA, and in this biosynthesis process, there are 2 enzymes that play important roles. First is TAA and then YUC. At 1 millimolar L-tryptophan group, we supply enough substrate to boost the IAA production, so the stem elongate. But at the 3 millimolar group, 2 things go wrong. First, the YUC enzyme saturate, and this is a rate-limiting step. Second, and most importantly, high IAA triggers ethylene biosynthesis, and ethylene is a stress hormone that actively closes cell elongation window. So 3 millimolar group is actually worse. This figure illustrates the IAA ethylene crosstalk pathway. So the elevated IAA would activate the HCF pathway and relieve of transcriptor factors called ARF that induce ACS gene expression. So this help convert ACC into ethylene, resulting in physiological responses such as stomata closure, leaf reasoning, and root growth inhibition. Next, we propose hypothesis 2 and hypothesis 3, which tell the full story of plant death. Hypothesis 2 proposes that the observed plant death was caused by chronic waterlogging rather than by L-tryptophan treatment. As shown on the left side of the flowchart, excessive watering and continuous rainfall cause prolonged soil saturation, leading to oxygen depletion on the root zone. Without sufficient oxygen, root cells could no longer sustain aerobic respiration and ATP production. The roots therefore switch to anaerobic fermentation. Although this pathway also provides limited energy, it produced toxic bioproducts such as ethanol. As a result, ATP level declined, and root tissue became necrotic, and the root turned black. The damaged root was then more susceptible to pathogens such as Phytophthora and Phythium, which further accelerate root decay. Importantly, this waterlogging event occurred before any L-tryptophan treatment were applied, which is before our experiment. Therefore, the root damage is more likely an experimental artifact caused by excessive water availability rather than by L-tryptophan. Then, moving to hypothesis 3, we propose that the subsequent heat wave acted as a final trigger for the plant death, while UV radiation was not the primary cause. This is supported by the observation that outer side plants is actually exposed to higher UV, but yet they survived. As shown on the right side of the flowchart, temperature rises from the heat wave greatly increase water demand. However, as we mentioned, the root has already been damaged by waterlogging. They could not supply enough water to the shoots. As a result, stem transportation declined, and the stomata closed. Photosynthesis was reduced and leaf rapidly fell off. In summary, our evidence support a two-step process. Chronic waterlogging first damaged the root system, and subsequent heat wave trigger hydraulic collapse and plant death. And here's some references of the discussion section. Thank you.